Good morning to everybody. Today's topic in the chapter Microbial Cell Culture and its application is culture preservation and application of microbial cell culture. Okay. So what are the different method for culture preservation? That is agar slant methods. Agar slant method. In this method, we put the agar media in a test tube, in a bigger size test tube, and after pouring the liquid media we can give the tilt shape of that test tube, and when that media becomes solidified, then we streak on the surface inside the test tube the bacterial culture, and then we incubate for 1 days, and after growth of that culture we preserve at 10 degrees Celsius, 4 degrees Celsius, or minus 10 degrees Celsius for 6 months. Okay. So agar slant method, it is a simple methods for culture preservation, bacterial culture preservation. So simple agar media along with agar becomes solidified when it becomes solid at room state. Then the tilted area of the agar inside the test tube that contains some area, on that area we streak. We do the process of streaking, means with the help of inoculation loop we do the streaking for the further growth of that bacteria. When we incubate at room temperature for overnight, then we preserve at 4 degrees Celsius or minus 10 degrees Celsius. That is the agar slant method. Second methods. Second method for preservation. Second method for preservation of bacterial culture. So you know that is the agar slant culture covered with the mineral soil. Agar slant culture covered with mineral soil. So the same process we do. We take agar slant, and then yes, then we, after streaking the bacterial culture, we put a layer of that mineral soil so that bacteria will not grow unnecessarily at room temperature. So that is the second method, that is agar slant methods. Okay, covered with mineral oil. Okay. And the next method for culture preservation, next method for culture preservation, that is the storage in saline solution. What do you mean by saline? That is the 1% sodium chloride solution. So we put the agar, we put the bacterial strain inside the media that contain a 1% salt solution. So salt solution is bacteriostatic. It will prevent the growth of that bacteria in the media. So when we are going to store the bacterial culture, we add 1% the sodium chloride, that is the salt, and salt, that is 1% sodium chloride, means 1% means whatever the volume of the media, that is 100 ml, then we add 1 gram of sodium chloride. So that sodium chloride will prevent the growth of that bacteria at room temperature. So this is the third method, that is the storage in the saline suspension, and this is temporarily stop the growth. Yes. Any question? Any question? Uh, yes. Any question? Please stop talking. Please stop talking, please. Off your mic. So, why you are talking, please? Yes. So, next method for culture preservation is the storage in saline suspension. You know, 1% solution of sodium chloride that prevent the growth of that bacteria, because bacterial growth is bacterio means sodium chloride solution is bacteriostatic, so it prevents the growth of that bacteria. Okay, so third method, that is the storage in saline solution. Next method is preservation by drawing in vacuum. Preservation by drying in vacuum. How can you do? Culture are dried over calcium chloride in vacuum. Bacterial culture solution are dried over calcium chloride and then stored in refrigerator at -10 degree, -15 degree. So this is the method for longer period preservation, that is air dried culture. We are going to preserve for 1 year, 2 year, 3 year, like that. And one of the most important method, that is the cryopreservation. The cryopreservation, it is a method that is applicable both in microbial cell culture preservation, plant cell culture preservation, then animal cell culture preservation at -196 degrees Celsius. You know, so what is the method of cryopreservation? You take the bacterial culture and put in 10 to 30, you see in your book, 10 to 30% of glycerol or dimethyl sulfoxide. Glycerol or dimethyl sulfoxide. What is the percentage? 10 to 30%. Do you know what do you mean by 10 to 30%? Means if you are making 100 ml bacterial culture, then you have in the water, if you want to preserve that bacteria, then you have to prepare the solution 100 ml water, you add 10 grams of that glycerol or 10 gram or dimethyl sulfoxide or other cryoprotectant preservative, that is mannitol, sorbitol, like that. So this is the one of the most important method for longer duration, more than 5 year, 10 year, like that. If you want to preserve the bacterial culture at -176 degrees Celsius to -196 degrees Celsius. Why we are using this chemical? Because this chemical act as a cryoprotective agent. What do you mean by cryoprotective agent? Means a chemical that will prevent the formation of ice crystal inside the bacterial cell. That chemical is known as cryoprotective agent. Means if there is a formation of ice crystal of bacterial cell, you know, the same volume of ice and same volume of water, which have same amount of ice and same amount of water, which has higher volume. Ice have higher volume, so it will disrupt the bacterial cell wall and disrupt the identity and lose the identity. Bacteria will lose the identity. So that's why we prevent the formation of ice crystal. We prevent the formation of ice crystal inside the bacterial culture. Okay, so this is the cryoprotective agent, which is the most important agent that help in preservation of microbial culture or plant cell culture or animal cell culture at -176 degrees Celsius or -196 degrees Celsius. So one of the most important method for strain preservation, bacterial strain preservation, is storage the bacteria in liquid nitrogen. Inside the liquid nitrogen, what is the temperature? Minus 176 degrees Celsius or minus 196 degrees Celsius. So please off your mic. Okay, I'm requesting the parents or students, please off your mic. It is creating disturbance. Okay, so this is the one of the most important method for strain preservation is cryopreservation. Means in presence of cryoprotective agent, we are putting the bacterial strain at minus 176 degrees Celsius to minus 196 degrees Celsius in liquid nitrogen. So this is the temperature of liquid nitrogen, and we can easily maintain the bacterial strain for longer. After longer duration, we can use that same bacterial strain. So this is the one of the most important preservation method, that is cryopreservation. Next is lyophilization. Lyophilization or freeze drying, involving the freezing of culture followed by drying. First, you freeze the bacterial culture at minus 15 degrees Celsius, minus 196, and then result the formation, sublimation of cell water. Means after freezing at minus 10 degree or minus 15 degree, we are going to remove the water. You know sublimation, conversion of the solid ice directly into water vapor. You know that is known as sublimation. So we are going to remove the water content of the bacterial cell inside the, what is the water present inside the bacterial cell at minus 10 degrees Celsius, that is at low temperature, and lyophilized culture may remain viable for 5 to 10 years. How much year? 5 to 10 years. So this is the one of the most important preservation method, that is the lyophilization. First step is freeze drying. Means you are going to put the chemical at low temperature, and then whatever the water converted into ice directly converted into vapor form of the that ice. So that is known as sublimation. I think you have learned this word sublimation. That is the conversion of ice. That is the water first converted into ice at low temperature. In case of freezing of bacterial strain, then that ice directly converted into vapor without without going to water stage. So that is the lyophilization. And then lyophilized culture, that is in the powder form, it will viable for 5 to 10 years. So these are the strain preservation method. First is agar slant culture method. Second is agar slant culture covered with mineral oil. Third is storage in saline suspension. Fourth is preservation by drying in vacuum. Okay, and that with the help of calcium chloride solution. Then last, that is the cryopreservation, and ultimately one is lyophilization. So these are the different method for cryopreservation. Lyophilization or freeze drying, both are same. Simple process is, first you put the bacterial culture at very low temperature. In that situation, water present in the bacterial cell converted into ice, and then through vacuum drying we are going to convert the ice directly into water. Now come to the next topic, that is next topic is application of microbial cell culture. Okay, so before going to topic the application of microbial cell culture, you try to know culture collection center. You know, so there are national culture collection center and international culture collection center, also known as gene bank. Okay, so gene bank is the place where we are going to maintain or preserve the bacterial strain through the process of lyophilization through cryopreservation for 5 to 10 year and more than 10 year. Okay, so if you have to you you are going to do any experiment regarding the bacterial cell culture, so you will get the pure culture strain only from that gene bank. I think all of you have visited the gene bank, the bacterial cell culture in Microbial Institute of Technology, that is the MTech. So you have think the so many bacterial cell culture strain center, and one of the most important bacterial cell culture center, that is the Microbial Technology Chandigarh. It is written in your book, Institute of Microbial Technology Chandigarh, and you have visited this lab. And National Culture Center, named as National Bureau of Agriculture Important Microbes, that is present in the Mau, in the Uttar Pradesh. Mau district is there in Uttar Pradesh, but this MTech, that is the place where there is a gene bank for microbial cell culture. You can also take the sample for particular strain. So lot of strain are preserved here. The different bacterial strain are preserved here, and you have visit that the steps how they are preserving the bacterial cell culture. They have explained in a nice way. Okay, so this is the culture, and American type culture collection center, that is American type culture collection center in USA. NCIB, National Collection of Industrial Bacteria, that is in UK. And Dutch and Samlang, von Microorganism and Jewel Culture, Germany. So there are three places at international level. There is a gene bank that is going to utilize for research purpose. That is in USA, ATCC. So what do you mean by ATCC? American Type Culture Collection Center. You know, American Type Culture Collection Center. That is ATCC. And then second, that is National Collection of Industrial Bacteria, that is in UK. National Collection of Industrial Bacteria, that is in UK. So these are the culture collection center. And also in the Germany also, the other type, that is DSM, Dutchen Sam Samlang, and M for microorganism. So this is the present in that gene bank or culture collection center is present in Germany. So this is the international type gene bank or culture collection center. And you will find microbial technology Chandigarh, Indian culture collection center, and NBAIM, National Bureau of Agriculture Important Culture, means that is specific for agriculture important microbes. It can be used as biofertilizer or other purposes. Okay, so you must learn these examples. There are the international type culture collection center and national type culture collection center, where we are going to preserve. I have already discussed the gene bank in plant tissue culture. The same way we are going to maintain the bacterial cell culture or fungal cell culture or microbial cell culture, viral cell culture. So viral cell culture is present in Pune, and the bacterial cell culture, that is present in the that is in Chandigarh and in Mau, Uttar Pradesh. So these are the culture collection center all over the world and all over the India. Next, come to the application of microbial culture technology. What is the application of microbial culture technology? Okay, so try to know production of whole microbial cell. You know, food. That is the food for animals or food for the human beings. So if you will find the examples, that is the so many examples are there, like the Candida utilis and spirulina are used as a single cell protein. We can produce huge quantity of protein. What are the example? Single cell protein. That is the one of the most important example. That it is responsible for high quality protein production that can be used as a food for the poor man's, and it is also produced throughout the year with there is no effect of climates. for single cell protein production, that is going to produce by bacteria or algae, or you can find. So what are the different examples through which we are going to produce the single cell protein? SCP. You must remember SCP. It is written here. It is written here in your book. SCP. This is single cell protein. Yes, just see the cursor. Single cell protein. Okay. So the whole organism are used for production of the protein, that is the source for the for man people or the for animal also, and we are going to produce that protein easily throughout the year, and that is the that requires a very limited area, that do not require very large farm, and we can produce by using wastewater. So production help in reducing the water pollution. So there are so many advantages. It can reduce the water pollution. We can produce that protein through the wastewater with the use of that microorganism like spirulina, torula, candida. That is the candida utilis, that is the fungus. So these are generally used for huge quantity of production of protein, and there are so many advantages. There is no effect of environment during production. Second, we can reduce the water pollution through this production process by utilizing that microbes. So that is known as single cell protein, and that do not require large area also. Okay. Next example is vaccine production. You know vaccine, typhoid vaccine, Salmonella typhi, that is caused the disease tetanus, that is the caused by Clostridium tetani and tuberculosis. So there are so many vaccine are produced through the respective bacteria through this microbial cell culture. So you know the disease typhoid. Typhoid is caused by Salmonella typhi. And second, tetanus, Clostridium tetani. So that is the bacteria. And again, tuberculosis, also Mycobacterium tuberculosis. So these are the different type of disease caused by bacteria, and we are able to produce different type of vaccine. You know, this is the given in your book. Vaccine for typhoid, tuberculosis, that is used for precaution such as bacterial vaccine. So vaccine for typhoid and tuberculosis. Okay, we can easily produce through using whole organism, that we are going to attenuated the bacterial state. Attenuation means decrease the capacity of that bacteria, and the same organism of bacteria is used as a vaccine, means decrease the pathogen disease causing capacity of that bacteria, and whole organism that can be used inside the vaccine for production of antibody in any living organisms, in any human body. That is the concept of vaccine production, means we are going to attenuate means we are going to decrease the pathogenicity, means reduce the level up to that level, so that do not cause disease. And same bacterial cell of salmonella or mycobacterium that cause tuberculosis, and other clostridium that can cause tetany, that same bacteria is going to utilize for production of vaccine. So this is the application of whole microbial cell for production of food, that is the single cell protein, and vaccine for production of vaccine against the disease like typhoid, the disease like tetanus, the disease like tuberculosis. Next is the production of primary metabolite. You know, primary metabolite and secondary metabolites. So primary metabolite, these are the alcohol production, acid production, then vitamins. These are the produced through exponential growth phase. Primary metabolite are produced through exponential growth phase. You know exponential growth phase, that is the different stage of the bacterial growth, that is the lag stage, log phase stage, stationary phase stage. So generally we are going to produce the compound like alcohol, like acid, like vitamins like vitamin C or citric acid or vitamin B12. So these are the chemical that is going to produce through the exponential growth phase, that is the log phase. You know, I think you all of you have seen the exponential growth phase. That is a concept for production of primary metabolites. So with the help of that use of that bacteria through fermentation, with the help of so many bacteria involved for production of different type of chemical compound. So it is listed in your book. If you see the vitamin B12 propionobacterium or many, you know, it is given in your textbook. I'm talking about the textbook. In your textbook, it is written propionobacterium, that is responsible for vitamin B12 production, and vitamin C, ascorbic acid, that is the also known as ascorbic acid. So you can produce that vitamin C through bacteria. You know, citric acid, aspergillus niger, you can also produce the citric acid, that is the fungus is responsible for production of citric acid. So it is give, I think it is given in the in your book. You see, this is the citric acid, aspergillus niger, Niger, and then you will find vitamin B12, propionobacterium ceremani. So this is the bacteria. Through the fermentation inside the fermenter, we are able to produce that compound. It can be intracellular, it can synthesize either inside the body or extracellular. I have already discussed these things. So these are the responsible for production of different type of chemicals like citric acid, alcohol, you know, yeast, Saccharomyces cerevisiae. Then uh, you will find the interferon. You can produce the interferon, the insulin, the growth hormone from the Escherichia coli. That is the most important common bacteria that is going to produce that useful compound, that is insulin for treatment of diabetes, and growth hormone for the enlarging the length height of the individual, and then interferon, that is a protein, that is gamma interferon, that is used for treatment of the disease like cancer. Next is hepatitis B vaccine antigen. It can also produced by yeast, that is Saccharomyces cerevisiae. And next is, that is the biodegradable plastics. You have to remember that example. Biodegradable plastic, PHE, polyhydroxybutyrate. This is the unit for making biodegradable plastic. We can produce that biodegradable plastic that is eco-friendly, and we can produce that biodegradable plastic material, that is polyhydroxybutyrate, from the alkaligens eutrophus. So these are the different examples through which we can produce so many chemical useful compound microbes, through which we are going to produce so many useful compound. You know, dextron, one of the most important component in your syrup, that is the drugs syrup you are taking during cough. So leuconostic mesenteroid, it is also bacteria. Amylase, that is also used for digestive ad syrup. So aspergillus oryzae, that is the fungus. So you try to know who is the fungus and who is the bacteria. This is the fungus, Saccharomyces cerevisiae. This is the fungus also. Again, this is the penicillium. I have already discussed this Penicillium notatum is the initial strain for producing penicillin, but this is the commercial strain through treatment of mutagen, and through recombinant DNA technology. That is the production of that strain. Then next is bacteria, the Streptomyces griseus, that is streptomycin, that is the antibiotics. So these are antibiotics, penicillin and streptomycin, but these are the chemical that is used for different purposes. Next is L-lysine, that is amino acid, one of the most important amino acid for treatment of disease, that is the tonic. Vitamin B12, you know, vitamin B12 generally present in all the tonics, generally vitamin D, vitamin B12. So generally you can produce the propionobacterium from the propionobacterium. So generally people are going for testing the vitamin B12, vitamin D, and what is the source? That is the bacteria. You can produce the vitamin B12 in huge quantity from bacteria, that is propionobacteria. And amylase. Also, a digestive syrup can be utilizing this enzyme, that is the Aspergillus oryzae, that is the fungus from which we can produce that enzyme. And then dextron, I have already discussed that dextron is one of the most important constituent of what? Iska constituent hai syrup ka. Then insulin for diabetic person, growth hormones for that a person that they know growth occur, the height of that individual is very less. In that situation they are taking the growth hormone. So from where it is going to produce? From the bacteria, that is Escherichia coli. Next is interferon. So how they are going to produce? Through recombinant DNA technology. It is written here. No, it is written here through recombinant DNA technology. So try to know the example. This is the most important example for your CBSE exam point of view. Okay. Next is polyhydroxybutyrate. That is the chemical that is used for making biodegradable plastic. So there is lot of concept are there. Just clean plastic free environment. So there, that plastic, if you are going to utilize, that will degrade. Okay. Otherwise plastic is not going to degrade for millions years. Okay. So that can easily be degraded, and that is going to produce by bacteria alkaligens eutrophus. So everybody please try to know the what are the different chemical, and how they are going to produce this chemical, what is the source of that chemicals. Please everybody please try to know the concept. ठीक है, तो आपको पता होना चाहिए कौन कौन सा केमिकल किस बैक्टीरिया से प्रोड्यूस होता है। आपको बिल्कुल याद होना चाहिए। दैट टेबल इज वेरी वेरी इंपॉर्टेंट फॉर योर एग्जामिनेशन पॉइंट ऑफ व्यू। ओके, तो ये बताओ कि आवाज आ रही है? आर यू लिसनिंग माय साउंड? आवाज आ रही है? हाँ बिल्कुल। तो ये कोई बता सकता है कि विटामिन B12 किससे प्रोड्यूस होता है? विटामिन B12 किससे प्रोड्यूस होता है? कोई बताएगा?
प्रोपाइनोबैक्टीरियम।
प्रोपाइनोबैक्टीरियम, यस। ये आपको सिर्फ सीबीएसई में नहीं पूछेगा, ये आप अगर आप इंट्रेंस देंगे, किसका? मेडिकल इंट्रेंस, वहां भी पूछता है। इसीलिए मैं बता रहा हूँ। अभी किसने क्लास छोड़ा है? अभी मैं ये कहना चाह रहा हूँ कि बीच में क्लास होने की कौन सी इच्छा होती है? क्या हो गया? अभी कौन छोड़ रहा है क्लास? ये बताओ। अभी रिसेंटली कौन बाहर गया? बताइए। रोल नंबर 1, रोल नंबर 1 इज प्रेजेंट। शिल्पा, रोल नंबर 1 इज प्रेजेंट और नॉट? अभी तो थी। So what are the application of that microbial cell culture? These are the application, actual application. Next is antibiotic production, secondary metabolite. What do you mean by secondary metabolite? What do you mean by primary metabolite? So primary metabolite generally produced through the log phase, that is the exponential phase, that is the sigmoid phase. You are not understanding the what do you mean by lag phase, what do you mean by log phase. So try to know what is lag phase, what is log phase. You know this thing or not? I am explaining the lag phase, log phase. I have already explained one time. So just see, this is the growth of bacterial culture. If you see the in x-axis there is a time, in y-axis number of microbes. Then from the beginning, initially the bacteria is in its initial stage, that is the lag phase. There is no active phase. Although there is a growth occur, but growth of that bacteria is very less. If you are going to observe the log phase, growth rate is very high. So in last class I have discussed the slope tan theta, that is the angles. If you make that angle tan theta, you have learned this thing tan theta in maths in class 10 or like that. Tan theta, that is p by b, that is the some value. So angle is going to increase here. If you make the angle, that is going to increase here in log phase. It is gradually increasing, and the shape is sigmoids, S shape. Okay, so that is log phase, that is very active phase. When bacteria is entered in the log phase, then become very active phase. Only that phase there is a synthesis of primary metabolite like vitamins C, vitamin C, citric acid, or so many chemical compound like ethanols, then vitamin B12, that is going to synthesize in the log phase. But if you are observe the secondary metabolite, what is secondary metabolite? Secondary metabolite are toxin produced by bacteria, antibiotic produced by bacteria. So in which state it is synthesized? In the stationary state. Stationary means there is no active growth state. Means there is no increase in number. Number will maintain. Multiplication will occur. The bacteria multiplication will continue, but the number will not increase. So if you observe the slope tan theta, that remain constant throughout the stationary phase. So in that stage only there is a production of secondary metabolite. So what is the difference in primary metabolite? Primary metabolite are synthesized in log phase of the bacteria, and secondary metabolites going to synthesize in stationary phase of bacteria. What is the example of primary metabolite? Primary metabolite, that is vitamin B12, vitamin C, citric acid, ethanol, acetic acid. So these are the different chemical are going to synthesize actively by bacterial strain. That is, the bacteria is in highly active stage, in the highly dividing stage. So that is the log stage. Only we can going to produce the primary metabolite, and in the secondary metabolite production are occur only in stationary phase. That is the there is a no slope. That is the zero slope. The slope is going to increase, value is going to, angle is going to increase, that is the tan theta. So if you I have explained this thing, what is tan theta, and on the basis of tan theta, that the slope we can decide the bacteria is in which state. If the tan theta, that is slope is very less, then it is in lag phase. Slope is going to increase, then log phase, and slope is going to constant zero, then stationary phase. And when the number of population of bacteria is going to decrease, then it is the death. So these are the different stages. Okay, so you will learn these things, and also production of useful food, that is TAMP in the that is the fungal food in Japan, Indonesia, and we can also produce this stamp in so many Asian country. Next is biofertilizer. We can also produce biofertilizer from bacteria like Pseudomonas bacteria, Rhizobacterium bacteria. So these are the application part of the microbial cell culture. Okay. Next is biotransformation. You are going to transform the raw material into more useful chemical compound. That is also biotransformation. And biomining, extraction of metal like silver, like gold, with the help of bacteria, that is known as biomine, and production of recombinant protein. So these are the different chemical you are going to produce through the bacteria. So you all of you must write down the example for the production of different chemical compound like single cell protein, vaccine, temp, biofertilizer, primary metabolite, secondary metabolite, biotransformation, then biomining, bioremediation, and production of recombinant protein. So please, all of you must learn the examples for each type. Okay. So today you must go through the book and try to learn the examples and write down the application of biotechnology, microbial cell culture technology. Okay. Thank you.